Hi guys, welcome back to our podcast. I'm Serena.
I'm Sharvi.
And I'm Shanti.
And today we're going to be talking about epigenetic reprogramming. So epigenetic reprogramming is a biological process that changes how genes are turned on or off without altering the DNA sequence itself. It works by modifying chemical markers on DNA and proteins called histones, which control how tightly DNA is packed inside cells. These changes influence how cells develop, function, and respond to their environment. Scientists discovered that mature, specialized cells can sometimes be quote-unquote reset to be more flexible, stem-like state and through reprogramming, and this ability has helped researchers better understand aging, disease, and human development. Epigenetic reprogramming also plays a major role in regenerative medicine and stem cell research, and overall, it offers promising opportunities for treating illnesses by restoring healthy gene activity.
Epigenetic reprogramming happens at key life stages. So during gamete formation, which is like the sperm and the egg, many of the epigenetic markers are erased and reset. And then after fertilization, the embryo undergoes widespread demethylation and remethylation, which basically establishes a new developmental program. So some marks escape erasure, which raises questions about intergenerational inheritance. So basically, some of the main mechanisms include DNA methylation, which is where small methyl groups attach to DNA at these sites called CPG sites, and usually they silence genes by preventing transcription of the machinery from accessing them. Also, another mechanism is histone modifications. So basically, DNA wraps around histone proteins, and chemical changes like acetylation or methylation to these histones can loosen or tighten DNA packaging, which basically just overall affects gene accessibility. And then also non-coding RNAs, so basically small RNA molecules that can regulate gene expression by interfering with mRNA or modifying chromatin structure.
Okay, so one way that epigenetic reprogramming is used in your body is through rejuvenation. So an example of this is children are born young as babies, even though when they get their genes from their parents are technically old, and this is because of cellular reprogramming that happens biologically. And an interesting thing is that scientists can try to replicate this in a lab setting, which they think could eventually lead to something that could reverse aging. Okay, so basically how it works is, as Sharvi was saying, DNA methylation acts as a biological clock because these methyl groups are added to DNA as people age, and those methyl groups, as you age, their DNA methylation patterns become disorganized. So some areas have a lot of the methylation and some areas don't have enough. And this is bad because increased methylation in certain genes can hinder certain protective functions, which causes cells to lose their youthful identity and functionality. And so removing the methylation can allow cells to regain their youthful gene expression. And a fun fact is that researchers have developed specific methylation clocks that use specific DNA methylation patterns to predict an organism's biological age, and it's actually super precise, and it's a highly used way to find the age of something that you don't know, which I think is really interesting.
Also, indices of normal epigenetic programming can contribute to cancer, neurological disorders, and autoimmune conditions. So, for example, hypermethylation of a tumor suppressor genes can silence protective pathways, while global hypomethylation can increase genomic instability.
So some pros and cons for epigenetic reprogramming. So some pros include it can restore normal function in damaged or diseased cells. It reduces the need for organ transplants. It may lead to personalized medicine treatments. It helps advance stem cell and regenerative research and offers new approaches to currently incurable diseases. But some cons are that there are risks of uncontrolled cell growth or tumors, Long-term effects are still unknown. There are ethical concerns about human cell modifications, and techniques are complex and difficult to control. And lastly, not all cells respond equally to reprogramming. So I think that concludes today's episode. I'm Serena.
I'm Shanti.
And I'm Sharvi.
Just a disclaimer, this podcast was researched and produced entirely by high school students, so not everything will be completely 100% true. Thank you for listening.
Thank you.